Good morning. It's a great pleasure to be with you here this morning. I have many people to thank for the ability to be here this morning. These include the wonderful people at Parks Victoria, John Sr., Mark Stone, many people at the Nature Conservancy Australia, Annabelle Robertson, Zoe Davis, Michael Looker, Thomas Foundation, Max Bork. Distinguished guests, friends, colleagues, ladies and gentlemen, I am really quite honored to be here with you today in Melbourne and to deliver a keynote address here at the Healthy Park, Healthy People Congress. To date, I think the events of this Congress that I've observed reveal that what is going on here is extraordinary. The confluence of ideas and commonality of vision as to how protected areas may serve humanity has captured a moment, perhaps a tipping point, in the acceptance and incorporation of these vital ideas into this world. Today I'm here to share with you some of the work I, along with 100, and in fact several more scientists, have engaged in over the past decade that illustrates how our health depends on nature, and demonstrate how protected areas have a key role in caring for the countries that care for us. One such example are the two species of gastric brooding frog, pictured here. Now, two species of gastric brooding frog were discovered in rainforests in Queensland, Australia. The female swallows her eggs, where they fertilized eggs, which gestate in her stomach. They develop into tadpoles in her stomach, and they are born into the world through vomiting, pictured here, where they grow up into adults. Now, all vertebrates secrete acid and digestive enzymes in their stomach, and in fact, they secrete other substances that force food contents from the stomach into the intestines, including these frogs. And yet these frogs don't digest their young. It was discovered that these tadpoles of this species secreted a substance, or perhaps substances, that inhibited the digestive process and prevented the stomach from emptying. Substances that may have been useful to the treatment of peptic ulcer disease, a disease that perhaps afflicts as many as 1 million Australians and over 25 million Americans. But the studies on these tadpoles had to be stopped because, in fact, both species of gastric brooding frog went extinct. And the treatments that may have been made available based upon studies of these frogs have been lost forever. Now, I start with this disheartening example, not to depress you first thing in the morning, although perhaps I have, but because I think it illustrates a couple of important points for the talk I'm going to be giving. First, biodiversity, or the variety of life on Earth, is in trouble. Species extinctions are occurring at a rapid pace. In fact, 1/3 of all amphibian species are endangered with extinction. The second is that these frogs, these gastric brooding frogs in Queensland, in fact, many of portions of their habitat were in protected areas. But in fact, the thing that put them into extinction is likely a fungal infection. And that, in fact, we have several examples now of species that are on the verge of extinction or have gone extinct, where they've been in protected lands, but have succumbed to external influence. So this is but one insight into what biodiversity may mean for our health. But I get ahead of myself. Let me begin here. Although there's a substantial amount of controversy about the circumstances in which it was said and about the exact wording of the original remark, the eminent biologist J.B.S. Haldane, when asked what one could conclude about the creator from studying his work, is reputed to have said he had an inordinate fondness for beetles. And with good reason. There are some 350,000 described beetle species, comprising about 40% of all known insects, a number that is 6 times greater than all the vertebrate species that have been identified. This image is from a page of a British folio from a British expedition to Mexico in Central America from 1881 to 1884. It depicts 30 species of beetle, mostly from the genus Levia, each painstakingly drawn by hand in full color. We use species as one measure of biodiversity, but biodiversity is, of course, more than just the measure of species richness. It is the sum total of life on Earth, including the sum total of all the genes, species, local populations of individual species, and ecosystems. We use species because we can count them and because we're familiar with them. However, I must say, when it comes to our health, species richness is certainly perhaps not the best measure. We know about 2 million species well enough to have given them scientific names, but the total number of species may be 5 times that, if not more. To begin our tour of the natural world and its value to human well-being, let's begin here. This bacteria was originally discovered in a sulfur-containing hot spring in the United States. The hot spring, of course, gets its heat from deep within the Earth's crust. And this microbe may in fact be the most famous microbe on Earth, although I doubt you recognize it by its appearance. It was discovered in a place called Mushroom Spring by Thomas Brock and his then grad student Hudson Freeze in 1966. And this microbe's DNA polymerase, which is the enzyme that it uses to replicate its genome, has been harnessed for arguably the most powerful research and diagnostic tool in modern biomedical science, what's known as the polymerase chain reaction, or PCR. That's a picture of Mushroom Spring. Now, what PCR does is it takes a single piece of DNA and it makes lots of copies of it. And in order to make lots of copies of DNA, you need to separate the strands of DNA, as is imaged here. You need to use some heat to get the strands apart. Here it says 95 degrees Celsius of heat. Of course, that's a temperature that would turn everyone in this room into dinner, but for Thermus aquaticus, in fact, the temperature of the hot spring it lives in was about 70 degrees, so 95 turns out to be just fine. PCR uses this enzyme, this is the polymerase from Thermus aquaticus, to essentially fill in the gaps of genes. And when it's completed, you get multiple copies. And it's an iterative cycle so that you can repeat this process time and time again, taking very small amounts of DNA and making millions. Now, many of you are probably most familiar with this technique from having watched crime shows in which you're trying to identify the person at a scene of a crime. You find a piece of hair on the ground, they get some DNA off that, and lo and behold, you can make millions of copies and use that DNA to figure out who it is. As a scientist, I can tell you that we use it somewhat differently. This is a picture of what's known as a DNA microarray. Some artists have, in fact, seen pictures of this, and there's a whole art movement that is based upon images such as this, and they're quite elegant in their own way. But what this is one way in which the PCR technology has revolutionized the world of molecular biology. What's going on in this picture is that individual genes isolated from prostate cancer cell lines have been inserted into little divots in the surface of a microchip called wells. That's what each one of these dots is. Patients with prostate cancer who have biopsies have their cells digested, the genomes taken out, and the genes get labeled with fluorescent markers. And those pieces of the prostate cancer genome are washed over the surface of this microarray. And when genes bind, they glow. There are approximately, on this chip, 500 different genes represented from prostate cancer cell lines. Each one of these individual genes, the binding that occurs here individually when I was starting medical school would have taken a graduate student several weeks, so that it would have taken hundreds of weeks to do each one of these assays individually. Now with this technology, we can do 500 assays in a couple days. PCR has not only accelerated the pace of molecular biology, but it has given us the ability to get much broader and comprehensive pictures of vast amounts of biology, as you can appreciate here. Now, the one thing I didn't tell you about Mushroom Spring is that Mushroom Spring is in fact in Yellowstone National Park. This is a picture of the main gate to Yellowstone, for those of you who have not been there. Yellowstone, of course, the world's first national park. The main gate reads, "For the benefit and enjoyment of the people." Little did the park founders know how right they were.